Will nanomedicine take us to the age of immortality? Well, the word nano signifies a number, 10 to the negative ninth power, or 1/1000th of a meter. So trying to achieve precision at that scale is no walk in the park. For the longest time in human history, the way we made use of materials was limited to what we could see with microscopes or touch with our hands. Until one day, physicist Richard Feynman, with his talk, "There's Plenty of Room at the Bottom," suggested the crazy idea that we should construct things from the most fundamental building blocks of chemistry: atoms. Today, Feynman's ideas have finally come to life through graphene manufacturing, carbon nanotubes, and DNA origami that we can shape into nanobots. Furthermore, when it comes to applying technology towards extending the human lifespan, we realize that some problems we have in modern biotechnology could potentially be solved more quickly with nanotechnology. Unlike biotech, nanotech promises to take the manufacturing process to the level of atoms building molecules, rather than molecules building cells. The idea of nanomedicine, as defined by nanoengineer Ralph Merkle, is the use of atomic and molecular scale devices to repair damage in biological systems, adapting the precision used by synthetic chemists into the realm of medicine. This concept can take many forms, like nanomagnets, nanopores, nanoparticles, or buckyballs. First, we have the idea of nanomagnets, or artificial spleens, nanoscale beads that could clean out our blood and filter out antibiotic-resistant pathogens from our bodies. In the mainstream news, you may have heard of something called superbugs, microbes in the first world that have had their evolution accelerated by antibiotics overuse, excessive hand sanitizer, and excessive cleanliness. Their numbers are growing because sick patients tend to throw out their half-finished bottle of penicillin pills the moment they start feeling better. But since they didn't finish that treatment, some of the bacteria survive, and what doesn't kill them makes them stronger. These new veteran superbugs can no longer be killed with antibiotics anymore, potentially leaving nanotechnology as the only remaining way to get rid of them. A group of researchers at the Wise Institute at Harvard have attached magnetic nanobeads to mannose binding lectin, a protein associated with our immune response to bacteria, fungi, viruses, and parasites. These researchers created a stable form of the protein that does not cause blood clotting, and they used it in a kind of microfluid chamber meant to mimic the human spleen. Here's how it works. The nanomagnets bind to the mannose on the surface of the pathogen membranes, pulling the pathogens out of the patient's blood while it runs through the device. When the blood is pumped back into the patient, it leaves behind the viruses, bacteria, and parasites, which could then be analyzed in the lab. To see how well this device worked, the Harvard scientists infected rats with E. coli bacteria, let it sit, and then ran their blood through the artificial spleen. In the control group, which had no nanomagnets, only 17% of the rats survived, while in the experimental group that did receive the treatment, the survival rate was over 90%. Next, we have DNA nanopores. In the age of nanomedicine, biology will become nothing more than a regular computational system, and, in transhumanist terms, the biotic will no longer have any sacred distinction from the abiotic. As the famous Nobel laureate David Baltimore once said, all medicine will inevitably be reduced to computer science. Granted, biology is far more sophisticated than any other computer available to us today, but we're slowly starting to learn how to read and write DNA, just as we would with computer code. A group of researchers at the Institute of Electrical and Electronics Engineers have now taken the first step towards cheap and practical DNA sequencing. Last year, the researchers were able to use something called nanopores, tiny holes inside of a nanomembrane, just big enough to allow single molecules of DNA or protein chains to pass through and be altered, allowing us to not only read, but also write in the language of biology. By simplifying the methodological ability to sequence single-cell DNA, these nanopores are able to create a new age of submolecular analysis to better understand the genes that cause aging. Dr. Chad Merkin, a chemist in Northwestern University, has developed a new assay that reads the letters of DNA using gold and silver nanoparticles, while flagging variations of DNA that are sensitive to mutations. Current DNA sequencing is very expensive and time-consuming, but this diagnostic tool takes less than 2 hours and could be done in a single doctor's visit. If we could ever incorporate these nanopores into tiny robots, then future nanomachines might one day hang out in your blood, your gut, and probably even your toilet, where they will capture DNA fragments from cancer cells, identify cancer, and tell you if you are in danger. This technology would have been very useful to Steve Jobs, who actually had a 20-year window to get rid of his pancreatic tumor. The only problem was he didn't know about it. It's probably also conceivable that the toilet of the future will also talk to you, tell you if you eat too much, smoke too much, drink too much, and even give you personalized dietary advice to maximally extend your lifespan. Nanomedicine doesn't just stop at detecting cancer, but also treating it at the molecular level, using what you could call nanoparticles. One example would be photothermal metallic nanostructures tagged with cancer-seeking molecules. These nanoparticles can be injected into a patient, ingested into cancer cells, and then heated up with a giant external magnet, causing them to spin around and burn up the cell that ingested them. It sounds nice and fancy, but why is this necessary? Well, one issue we have with the chemotherapy we use today is that it's very harmful to healthy cells, causing bruising, hair loss, and extreme pain in the patient. Chemo doesn't distinguish between cancer cells and healthy cells. All we do is just pump it into the vicinity of the tumor and hope for the best. In the case of brain cancer, there's often nothing we can do. But now, MIT scientists are trying to change this, managing to treat a brain cancer called glioblastoma, using only 100 nanometer wide drug-containing capsules called liposomes. These liposomes deliver 2 drugs, temozolomide, which attacks the DNA of cancer cells, and bromodomain inhibitors, which prevents tumor regrowth. What makes liposomes special is that they're coated in a protein called transferrin, which latches onto the surface proteins of only cancer cells, leaving the healthy cells alone. Transferrin is also what allows these nanoparticles to pass through the blood-brain barrier and shield the 2 drugs from the immune system, as well as any hazardous molecules in our bloodstream that could prevent drug delivery. Liposomes are proof of concept that we can now get nanotechnology into the brain. Another team led by Dr. Dean Ho has developed even smaller nanoparticles called nanodiamonds. Diamond particles about 4 to 5 nanometers wide, coated with receptors that seek out the triangular-shaped proteins of tumor cells. But it'd be nice if we could get cancer-killing nanoparticles even smaller, so that they can get to those hard-to-reach places. Enter buckybalms. Nanotechnology would not be where it is today without the discovery of Buckminster fullerene, also known as the buckyball. It's an extremely stable arrangement of 60 carbon atoms in a cage-like structure, kind of like a soccer ball. It's a very strange-looking molecule, and we decided to name it after Richard Buckminster Fuller, the architect who popularized the geodesic dome. Fullerenes were discovered way back in 1985 by Harold Crotto, James R. Heath, and Richard Smalley, who isolated the carbon allotrope by firing lasers into graphite layers. Because it has special chemical properties, this molecule can have promising applications in nanomedicine that would allow us to deliver medications to infected cells, or even be used as a bomb. When a buckyball molecule is synthesized with 12 molecules of nitrous oxide, it can be heated up from the outside and made to explode inside individual cells. Definitely a clever idea. These are just 4 treatments, but there are many more still to come. The idea of nanotechnology may sound ridiculous, but to understand it, we need to remember that all living things, including ourselves, were once manufactured from individual molecules that self-assembled themselves without the intervention of production tools. Aging deconstructs us at the molecular level, so likewise, we'll need to reconstruct ourselves at the molecular level. Biotechnology can delay aging, but it can't hold it back forever, and nanotech will become the new biotech. It will be the sign of a radical new age still to come, and a new way to reprogram reality. But that is a topic for another time. Thanks for watching, and may you live forever, or die trying.